Okay, let's talk about gram-negative lab algorithm. Gram-negative means a bacteria has a specific cell wall structure that makes it stain pink or red on gram stain. Gram-negative bacteria have thin peptidoglycan layer, so it doesn't hold crystal violet well. Outer membrane on top of that, and the outer membrane contains LPS, lipopolysaccharide. Lipid A part is endotoxin, which can trigger fever, shock. And there are porins in the outer membrane. Those are tiny channels that let small molecules in. So why they stain pink? In gram staining, crystal violet plus iodine initially stain everything purple. And then alcohol or acetone washes out stain from gram-negative because peptidoglycan is thin and the outer membrane gets disrupted. Then they take up the counter stain, safranin, and turns pink or red. So gram-negative bacteria are usually more resistant to many antibiotics because of the outer membrane. Outer membrane blocks the entry of the antibiotics. And endotoxin, LPS, can cause strong inflammation causing sepsis, shock. So they often cause serious hospital infections depending on the organism. Okay, the lab pathway for gram-negative bacteria usually starts with one question: what is the shape on gram stain? So let's start with gram-negative diplococci. Diplococci are round bacteria, cocci, arranged in pairs like two adjacent spheres, often kidney bean-shaped pairs for Neisseria. So if there is gram-negative diplococci, think Neisseria and sometimes Moraxella. The key test to differentiate those is maltose fermentation. If maltose fermentation negative, Neisseria gonorrhoeae and Moraxella. If maltose fermentation positive, Neisseria meningitidis. So we can memorize as Meningo munches maltose, gonorrhoeae does not. Meningo munches maltose means Neisseria meningitidis is maltose fermentation positive, but Neisseria gonorrhoeae is not. Now, let's go to gram-negative coccobacilli. These are short plump rods. The classic list is very testable. I have a very nice mnemonic for this list. The mnemonic goes like, hey, Bruce acts badly at border pass in France. Hey, Bruce acts badly at border pass in France. Hey is Haemophilus influenzae. Bruce is Brucella. Acts badly, Acinetobacter baumannii. At border is Bordetella pertussis. Pass is Pasturella. In France is Francisella tularensis. Okay, so hey, Bruce acts badly at border pass in France. Haemophilus influenzae, Brucella acts badly, Acinetobacter baumannii. Border, Bordetella pertussis. Pass, Pasturella. France, Francisella tularensis. Hey, Bruce acts badly at border pass in France. It's gram-negative coccobacilli. Then next, we go to gram-negative curved rods. If it looks like a comma or seagull wing, they are gram-negative calf rods. The calf rods are typically oxidase positive. If it grows at 42 degrees Celsius, Campylobacter jejuni. Hot growth, inflammatory diarrhea. It has hot growth, which is growing at 42 degrees Celsius. Think about Campylobacter jejuni, which cause inflammatory diarrhea. If it grows in alkaline media, think about Vibrio cholerae. It's alkaline lover, watery rice water diarrhea. Okay, so Vibrio cholerae is alkaline lover, grows in alkaline media. If it produces urease, of course it's Helicobacter pylori. Helicobacter pylori produces urease, mixed ammonia, and it causes ulcers. So gram-negative curved rods are Campylobacter jejuni, which grows at 42 degrees Celsius, Vibrio cholerae, which grows in alkaline media, and Helicobacter pylori, which produces urease. Next, we go to gram-negative bacilli, which are straight rods. To differentiate those, we look at lactose fermentation. This is commonly tested with McConkey agar. Lactose fermenters turn colonies pink. Non-lactose fermenters stay colorless. If it is lactose fermenter, then split into fast versus slow fermenters. Fast lactose fermenters are EKE, E. coli, Klebsiella, and Enterobacter. Slow lactose fermenters are Citrobacter and Sriracha. Okay, so fast lactose fermenters are EKE, E. coli, Klebsiella, Enterobacter. Slow lactose fermenters are Citrobacter and Sriracha. Okay, next we talk about non-lactose fermenters. For non-lactose fermenters, we use oxidase test. Oxidase test is basically a quick do you use an aerobic electron transport chain test. The classic non-fermenting gram-negative rods, especially Pseudomonas and Burkholderia, are strict aerobes and rely heavily on oxidative respiration, so they are oxidase positive. Pseudomonas and Burkholderia are oxidase positive. If it is oxidase negative, we check with TSI agar for H2S production. TSI agar stands for triple sugar ion agar. If a bug produces H2S, hydrogen sulfide gas, the gas reacts with iron in the medium like ferrous sulfate and forms black iron sulfide. So if it is H2S positive, it will turn black on TSI agar. They are Salmonella and Proteus. If it is H2S negative, no black color will be seen on the TSI agar. They are Shigella and Yersinia. Okay, so H2S positive is Salmonella and Proteus. H2S negative is Shigella and Yersinia. Remember that Yersinia, although it's in the Bacilli group, it is often described as a pleomorphic rod or Coccobacillus. Okay, let's summarize quickly for the whole algorithm. Gram-negative bacteria shows pink color on gram stain. We differentiate them with the shape. First, Diplococci. Diplococci are all aerobic. We differentiate them with maltose fermentation. If maltose fermentation negative, Neisseria gonorrhoeae, Moraxella. If maltose fermentation positive, Neisseria meningitidis. Another shape is Coccobacilli. Coccobacilli, we remember, hey, bruise eggs badly at border pass in France. Haemophilus influenzae, Brucella, Acetylobacter baumannii, Bordetella pertussis, Pasturella, Francisella tularensis. And then there are curved rods. Curved rods are oxidase positive. If it grows in 42 degrees Celsius hot growth, Campylobacter jejuni. If it grows in alkaline media, it's Vibrio cholerae. If it produces urease, Helicobacter pylori. The last one is Bacilli. Bacilli, we look at lactose fermentation. If it ferments lactose, we look at whether it's fast or slow. Fast lactose fermenters are EKE, E. coli, Klebsiella, Enterobacter. Slow lactose fermenters are Citrobacter and Sriracha. Okay, if it is lactose fermentation negative, we look at oxidase. Oxidase positive or oxidase negative. Oxidase positive, Pseudomonas, Burkholderia. Oxidase negative, we look at H2S production on TSI agar. If there is H2S production, Salmonella Proteus. H2S production negative, Shigella Yersinia. Okay, that's it for gram-negative lab algorithm. Have a very nice day and best of luck.